I'm Alan Beggs. Maybe we'll just quickly go down the row here and introduce ourselves, and then I'll tell you a little bit about how this session will work.
My name's Casie Genetti. I'm a genetic counselor working with Alan on his congenital myopathy and rare disease research at Boston Children's.
I'm Chris Tan. I'm a genetic counselor. I work at Invitae, which is a genetics lab based in San Francisco.
Hi, I'm Soma. I'm at the University of Chicago, and I direct the molecular diagnostic lab there that does testing for congenital muscle diseases.
Hi, I'm Jody Westbrook, and I work at Invitae with Chris. I'm a scientist.
And I'm Sandra Donkervoort. I'm a genetic counselor working with Dr. Karsten Banneman at the NIH in the diagnostic research program.
Okay, so we are going to start with a little basics. Genetics is obviously a complex topic. It's something that not everybody is familiar with. There's a lot of terminology that's used. And so we're going to have Casie Genetti and Christopher Tan, two licensed and certified genetic counselors, give us a brief presentation, kind of put us all on, did I get that right or not?
No, that's right.
Sure. And put us all on the same page about some of the terminology. And then after that, we hope to open it up to questions. I know many of you, not everybody here, has a genetic diagnosis. So some of you may have questions about how you get a genetic diagnosis. Others may have questions about what it means once you get one. You can discuss questions about what is a VOUS or a VOUS, and other questions that you may have about this topic. So I'll turn it over to Casie.
Do I click with this? Okay, can I stay seated? Sure. All right. Yeah, I'm good here. Okay, so we're going to do a really brief overview. For some of you, this may be a lot of review, but we'll just get ourselves on the same page in terms of some of the terminology that we use. So just a little shout out to our National Society of Genetic Counselor for the slides. When we are talking about our DNA, it's good to just put everything into perspective. So inside of all of our cells, which we have in our muscles, but every organ of our body, we have our chromosomes, which you can think of as kind of our little packages of DNA. Inside the chromosomes, we have our strands of DNA, which are made up of all of our genes. And you all know many of the names of the genes that we've been talking about this weekend. And really, that's kind of the focus of a lot of the genetic testing that we'll be doing and that Chris will be talking about. But a good analogy, as genetic counselors, we love analogies. We think of our whole genome, or all of our DNA, together as a book. Inside that book, we have chapters. You can think of those as the chromosomes, so little packages that make up the entire book or genome. And then our genes are much more specific. Those are the sentences. And our genes, each one tells our body different directions in terms of how to grow and develop. And a lot of our genes, when not working properly, can turn into human disease. So specifically, something you're hearing all weekend are what we call mutations or variants. So variants are just changes within the DNA. Mutation and variant oftentimes are used interchangeably, but we like to stick to the term variant because we know that for changes in DNA, not all of them are actually causing some kind of problem with the gene itself. So a variant is a little bit of a better word to use. And what a variant is just any change from what we consider the reference, or something that is different than the general population. We all have many variants in our DNA, and a lot goes into determining which variants are actually going to cause health issues. So Chris is going to talk more about how we determine that. But you can just think of those variants as spelling changes. So here if you take a regular sentence and you change the H in hall to a B, it turns into ball, which changes the ultimate meaning. So something that you may have been hearing a lot about are the types of genetic variants. So we use different terms to discuss kind of the different ways that these changes are occurring. So on top, we just have the original DNA sequence or sentence. You may have heard people talking about missense changes. These are just single spelling changes within a gene that don't necessarily change the remainder of the sequence. We have insertions, which would be adding one or more of those base pairs or a whole word, as this sentence would be. We have deletions, which in the same way kind of remove a portion of the gene. Again, that can be just a single base pair or a larger number of base pairs. Then we have a duplication, which again would just be a repetition of a certain portion of that gene. And then a nonsense mutation. Nonsense mutation is a change that actually truncates the entire gene or protein. And so what that means is the particular change ends that gene early. And in those cases, the remainder of the sequence is no longer there or functional. So another kind of term that you'll probably hear a lot is something called zygosity. And so we just wanted to quick define what the different types of zygosity are. So each of these little figures, does this have a pointer?
It should.
Okay, perfect. All right. So each of these little blue figures right here are representing chromosomes or alleles. And so we would have everyone has 2 copies of the vast majority of their genes. When someone is heterozygous, what's that's referring to is having one variant or a change on one copy. When we'll go into some inheritance models in a second, but when we talk about someone who's heterozygous, in certain cases, in dominant conditions, that could mean that they are actually affected with that condition. But in the case of recessive conditions, that would be referred to as a carrier, and I'll show that in a moment. When someone's homozygous, that means they have 2 of the same changes, 1 on each copy. And then we also have a compound heterozygote, which would be someone who has a changed copy or each copy is changed, but they're just 2 different variants. And then the last example is something called hemizygous, and this is a special one that refers to just X-linked conditions. Hemizygous means that there's a change on the X chromosome, but then that individual is a male, so they only have 1 X and they have a Y. So that's referred to as hemizygous, whereas a female who has 2 X's would be heterozygous. All right. And then before I turn it over to Chris, we're just going to do a quick review of the different inheritance patterns. For all of these that we're going over, we'll show you a pedigree up here. So pedigrees are really helpful for us when we take a family history to look at the pattern of a disease within a family. And so when we see people, maybe multiple people in each subsequent generation having a disorder, that's a clue to us that it's likely dominant inheritance. And so if you look down here at just a single family, when you have a parent who's affected with a dominant condition, there's a 50% chance that they would pass that on to their children. So if someone had 4 kids, the likelihood that 2 would be affected. When we talk about recessive inheritance, oftentimes when you look at the pedigree or the family tree, you have one affected individual, but you don't see any family history of that. And that can clue us into a couple things. We oftentimes think that it could be recessive, but it also could be that it's a brand new change in that child. In this case, it's a recessive change. And when we think of recessive conditions, we know that both the changes have to come, one from mom and one from dad. And those individuals are called carriers. Many of you may know about that or be a carrier yourself. Carriers are typically not affected with the health condition, and they're not expected to show symptoms over time. It's just that when 2 individuals are both carriers for the same condition, there's a 25% chance that they could have a child affected with that condition. And also a 50% chance that their kids could be carriers, and a 25% chance that they could have a child that didn't inherit either one of those changes. And then the last mode we'll talk about is X-linked. So as I mentioned before, X-linked has to do with the X chromosome. Females have 2 copies of the X chromosome, whereas males only have 1. They have an X and a Y. So when a woman has a change on her X chromosome, she's typically thought to be a carrier, and that's similar to kind of a recessive model. But when she passes that down to a son, they don't have that extra copy of the X chromosome to kind of help it out, and they typically show symptoms. And so you can see here, when we see patterns of males being affected and coming from related females who are unaffected, that usually clues us into X-linked inheritance. And you can see down here, when a female is a carrier, she can have daughters who are either carriers or completely unaffected, and then she can have sons who are either unaffected or affected. It's a 50% chance. All right, so I'm going to go ahead and pass the mic on to Chris, and he's going to talk about how these different variants.
So I'm briefly going to talk about the different types of results that you could get from a genetic test result, or genetic test in general. And in general, there are 5 different types of variant classifications that will be reported on a test result. And I'm going to talk about the sort of 3 main types of results. So the first being a positive result. So a positive result is basically a pathogenic or likely pathogenic disease-associated variant was identified, right? This typically confirms or establishes your diagnosis, and predictive genetic testing is now offered to other family members. On the other end of that spectrum is a negative test result. So typically a negative result means that no disease-associated variant was identified, right? So this does not rule out a suspected condition. It does not eliminate the possibility of having a hereditary condition, and predictive genetic testing is not typically available for other family members. The last bin is an uncertain result. So has anyone had a genetic test that came back with a result that was uncertain? All right, I'm starting to lose people here, so I need you guys to get like a second win here. No uncertain results. That's awesome. So that bin typically refers to that there isn't enough information to determine if that variant could cause disease. And this is typically called a variant of uncertain significance, abbreviated either VUS or VUS, depending on who you speak to. VUSs in general are not recommended for use in clinical decision making, and VUSs are typically not recommended for predictive testing in at-risk individuals. So how does a laboratory come up with these classifications for these variants? So this involves typically a careful review of the medical literature, to determine has this variant been seen before, have experimental laboratory studies been performed, re-review population data. So if that variant has been seen in lots of clearly unaffected individuals, one would make the assumption that that variant does not cause disease. In the absence of experimental laboratory studies, laboratories often use computer models to predict whether or not a variant could be disease-causing or not. And often laboratories request testing of other family members to help determine whether or not that variant segregates or is transmitted with disease in a family. It's also important to keep in mind that even after all these investigations, sometimes the lab is still not able to determine if a particular variant is likely to cause disease, and again, that is a variant of uncertain significance. So again, I want to leave plenty of time for questions, so I'm going to skip to my loo. And end with this very last slide about genetic counselors. For those of you who maybe aren't familiar with genetic counseling or seeing a genetic counselor is typically an individual who obtains a detailed family medical history and reviews information about inheritance patterns. We help to identify at-risk family members, discuss options for testing, review the benefits and limitations of genetic testing, coordinate and interpret genetic test results, and address psychosocial implications of test results. There are 3 awesome genetic counselors sitting up at the podium. There are some genetic counselors out here in the audience. If you have questions for us, feel free to find us and ask us. And again, feel free to visit the National Society of Genetic Counselors webpage, which is www.nsgc.org, to help locate a genetic counselor in your geographic area if you aren't currently set up with one. Thanks.
I think some of you must have questions. Can we have anybody, any thoughts or questions from somebody in the audience? Raise your hand.
Was wondering how important it is that someone who has a known CMD, that their parents have genetic counseling and identify the possible variants.
I mean, I think it's very important to get the complete picture of the whole family, to know exactly, to trace it through the family. And it also depends on the type of CMD, so what the diagnosis is. Is it the one that you expect to be recessive, or is it the type that started spontaneously, new in the patient? And all that can be clarified through parental testing.
In case the disease is recessive and the genetics indicates that it's heterozygous, have you guys seen symptoms of the disease appear in that case?
So since I'm standing here with the mic, I can start to address that. It depends. When you say it seems heterozygous, there's two possibilities. One is that exhaustive testing has been done and that we're certain there's only one variant, mutation or disease-causing variant on one chromosome. And we are discovering new things about diseases and these conditions, and occasionally a condition that we think is recessive, turns out there may be certain genetic variants that can operate in a dominant fashion. However, what's much more likely is the possibility that there's a second variant on the other chromosome that we have not been able to identify. And this is particularly true for several of the very large genes that we talk about, for the ryanodine receptor, for the nebulin gene in Nemelin myopathy, for the titin gene. These are very large and complex genes, and the current state of genetic testing only allows us to identify a certain proportion of the disease-causing variants. And so in a number of cases, we have individuals who have a medical condition that we believe should be associated with, for example, a mutation in the nebulin gene, and we find one conclusive mutation on one chromosome, we can't find the other, so we say this patient appears to be heterozygous, and yet in reality they probably have a second change we just can't find.
Could you go into a little depth about like mosaic genetics? I remember hearing a little bit about it, but I would love some more information.
Sure. I mean, mosaicism, you're going advanced genetics here in terms of genetic concepts. But typically, so every single one of our cells has our DNA. So if there's a mutation, we expect it to be in every single one of our cells, starting from conception, it's there. With mosaicism, what happened is that part of the cells are completely normal, and the mutation is present in only a selection of the cells. So instead of being present in every single one of your cells, a portion is normal and another portion has the genetic change. So typically that happens in dominant situations where one copy is enough to cause symptoms. And then with the mosaicism, it depends on what the mutation load is. So if more cells have two normal copies, we expect milder symptoms than if more cells have an abnormal copy. And that can be completely random. Some tissue, it can be random with the tissue. So some tissue can have more normal or less normal, randomly develop, throughout growth. Does that answer it? Okay.
And I should say that mosaicism it's a reflection of a very normal process. So of our 3 billion bases that make up our DNA, every time a cell replicates, there's an opportunity for a mistake to be made. And typically during spermatogenesis or oogenesis, when our eggs or sperm are formed, there's several de novo changes, new mutations or alterations that occur. And the vast majority of the time, these don't have any impact on us. So if you sequence our DNA and you sequence the DNA of our parents, all of us in this room have a small handful of changes that our parents didn't have. What mosaicism reflects is simply one of those changes that has occurred after the egg is fertilized and while the embryo is developing. And so in one of those cells, a mistake has occurred. So some of the cells of your body carry the result of that mistake and others don't. And as I said, our bodies are mosaics for all sorts of changes, but the vast majority of them don't have anything to do with the way we look or function. It's only once in a while when one of them affects a gene that's important for us that then it becomes a medical condition. So, more questions. Gustavo has a microphone that works. Is this why people can suddenly become symptomatic? Is mosaicism a reason why people suddenly become symptomatic? Generally not, because by the time you're born, the mosaicism is already present. So we'll let, who would like to take on disease onset and what causes later onset for certain conditions? Anybody? And depending on the biology of the condition, there are certain things that can become symptomatic later on. So there's a group of conditions, central nuclear myopathy, that my group is very interested in. And one of these forms, myotubular myopathy, affects a gene that's important from birth. And so virtually all the boys with myotubular myopathy are affected and weak and hypotonic right from birth. There's another gene called dynamin 2 that also causes a form of central nuclear myopathy. And individuals with dynamin 2 related myopathy often appear completely normal at birth, and it's only later in life. So the differences between these two conditions really is related to the biology of what that protein does.
I just want to add to that, there's still a lot that we don't understand when it comes to genetics and how it plays out in the family. Sometimes we see some variability in terms of disease onset and progression, or even within the same family, one patient may have more problems with contractures and the other may be more weak. So there are other factors at play that can change disease progression a little bit, but in terms of whether there is something that causes the onset of symptoms, there's no like environmental or exposure or anything that we know of that will like set off disease onset.
So Dr. Soma Das leads one of the premier genetic testing laboratories in the country at the University of Chicago and has been involved in the development of tests over many years. And I think many of you have undergone a bit of a diagnostic odyssey with tests that maybe initially asked about a single gene and a single mutation. Maybe later on you had something called a panel test. And maybe eventually some of you had whole exome sequencing. So I'm going to let her talk a little bit for a few minutes about the process and the evolution of these tests, and in particular what it means to get a negative result and what that doesn't mean, because in some cases, as we know, negative results are not so meaningful.
Thank you. So our knowledge has really increased. Our knowledge has really increased these past several years. So way back when, we would do testing for a single gene. Like if a patient is suspected to have myotubular myopathy, our lab, gosh, it was about 20 years ago now, or 17 years ago, we did testing for the gene, the MTM1 gene, which is known to be involved in myotubular myopathy. And so the test that we developed in the lab was to sequence that gene to look for a mutation. We now know that there's a lot of genetic heterogeneity, and there are a lot of genes that can result in similar phenotypes. So in myotubular myopathy, for example, it's not just the MTM1 gene. There are now, we know, several other genes that, when defective, can also result in myotubular myopathy, or phenotypes that resemble myotubular myopathy, right? And so as we get this information, as researchers are identifying these additional genes in the diagnostic lab, what we can do is we can develop more comprehensive tests so that we're now not just sequencing the MTM1 gene, we're sequencing the MTM1, the dynamin 2, the RYR1, the titin, so all the other genes that can also result in this sort of a broad phenotype. We now do testing for a panel of genes. So that's when now testing has gone from doing single gene testing to doing panel gene testing, where we're sequencing many genes at the same time. And there's a technology that the labs all use now. It's called next generation sequencing, which is a more high throughput method of sequencing these genes that allows us to sequence many more genes at the same time. So for example, in our lab, we have different panels for different subtypes of muscle disease. So we would have a congenital myopathy panel, we have a congenital muscular dystrophy panel, we have a congenital myasthenic syndrome panel, a limb-girdle muscular dystrophy panel, and these panels all contain genes that are associated with these different phenotypes, right? Although it's important to also understand that many of these genes that result in, say, congenital myopathy also does result sometimes in a phenotype of congenital myasthenic syndrome. So you can have the same gene on many different panels because they can result in different phenotypes. And on the other hand, labs, we have this too. We can have a much broader panel that we call like a neuromuscular panel that has a combination of all of these genes. For example, ours contains like 130 neuromuscular genes in one panel that now does sequencing for all these genes. If the physician is not, if the patient, if they're not quite clear that the patient has a particular subclass of muscle disease, they may want to just do testing for a much broader group of diseases, and so then that would be more beneficial to do a more broad, a larger panel testing. And then, of course, so with the lab, we do the sequencing and we analyze all the genes in the panel to see if we identify any mutations. And if there are often times that we don't, right? We've sequenced all of these genes, but we haven't found anything. It's a negative result. We haven't found a mutation in any of the genes that we have sequenced. Now, of course, what a negative result sometimes, it could be that the mutation is actually present in one of those genes. We didn't see it because in the lab, these genes can be very large. And in the lab, we are sequencing generally the coding region of the genes. These are the regions of the genes that's much easier to understand or interpret when we do find an abnormality, what it means. But it could be possible that there might be a mutation deep within an intron of the gene, which is a region of the gene that's non-coding that we don't sequence. It could be in the regulatory region of the gene that we're not sequencing. So a negative result doesn't always mean that it's truly negative for those genes that we tested for. I would say a large proportion are present in the regions that we sequence, but there are going to be some that we're going to miss because of the limitations of the technology. And then the next phase after that, so I talked about single gene testing, then there's panel testing, and now there's exome testing. And exome testing is when we're doing sequencing of all the genes in our system, right? So it's not just genes for neuromuscular disease, but it's all our genes. And this is a test that is done when a physician we may identify mutations in some other gene that's not clearly associated with neuromuscular disease. So we might find something that was unexpected. And for patients who have a much sort of a broader, non-specific phenotype, doing exome sequencing might be a way to go. I also want to say that though it's really important to do this testing in concert with genetic counseling, because when we do these tests, we can sometimes identify lots of different sequence changes, some of which might be the cause of the condition, and some are what we call these variants of unknown significance. We don't know what this sequence change means. And this is where it's really important to have these test results interpreted by a genetic counselor, because they can really explain and go into detail as to what all of this means. So based on the kind of condition or the situation, the physician will really decide what's the best kind of test to do, whether exome sequencing or single gene sequencing or panel testing, what might be the most appropriate. So, yeah.
Good. Well, thank you very much. Okay, we have one more online question, and then we'll look for one last question and then let you guys get some dessert. And this question online was from an individual with a COL6A2 mutation wanting to know, they're overseas, what kind of tips could we provide to get testing from overseas? And I think the first question is for other family members, I should say. The first question really is, who is it appropriate to get additional testing for? And this is really a question that your physician needs to think about and answer and discuss with you, because depending on the particular form of the condition that you have, if it's a childhood onset form and you have like an older aunt or somebody, there's not going to be much reason. And this is a family in Australia, thank you. And so It may not be appropriate to test everybody, and that's a decision that should be made with a physician. There are genetic testing labs around the country that can do this. Certainly within Australia, there's a very active one I know in Perth, in Netherlands, in Western Australia, run by Professor Nigel Lang. They can do COL6A2 mutation testing. There's also a group down in Melbourne and a group in Sydney. And then, in addition, there are labs here in the United States. There are both academic labs like the one at the University of Chicago you heard about, and in Vitae in San Francisco, and they will actually, I'll let Jody Westbrook tell you what they have to offer.
Well, we do accept samples internationally, as do many other labs in the U.S. It sounds like if you have other family members you're interested in getting tested, it's typically most efficient to do that through the lab that did the initial testing for the affected individual. Were there any more specific concerns about that online? Okay. Okay. Oh, okay.
We have one question in the back and then
So
in the back.
I think yes. There's, that's probably unrelated.
I have.
You cannot.
Yeah.
Hello.
Hi there. I have a question. Oh no, it's not working.
No. Hi, thank you. First of all, congratulations on a truly fabulous meeting. I'm a genetic counsellor from Melbourne, Australia, so I might be able to help the person inquiring online.
And Monique?
Yes, absolutely.
Is it working? No, it's not. Is it good? Hi there. I have a quick question. How do you determine a gene is pathogenic? Meaning it does relate to that disease. How do you make that mutation of that disease pathogenic?
Well, we look at a lot of different kinds of evidence. So if there's a mutation in a gene and we're trying to decide, is it causing disease? The first thing we do is see, is that mutation present in healthy individuals? And if it hasn't been seen in healthy individuals, that's one piece of evidence that it might be causative. Then we look for other genetic evidence. Has it been reported previously in other people who are affected with the same disease? And then we look for experimental evidence. Are there researchers who have done experiments about this specific mutation showing that it is detrimental to that gene? And it's really just dependent on how much information is out there. So when there's a gene that's not very well studied, you could find a really rare mutation that may very well be harmful, but there's just not information out there. And at that point, that's when it ends up in this bucket, this large bucket of things that we call VUSs, or variants of uncertain significance.
And in addition to that, on the clinical side, it's also important to see, does this fit with the symptoms? Like, does this variant in this gene fit with what we know clinically about this patient? And what's really tricky is we all have variants in our genetic information, so if we run genetic testing on everyone here, we all gonna end up with a whole bunch of variants. So there's gonna need to be clinical correlation to see if it all fits together. It's a complete picture.
Okay, so I think we have another session starting in a few minutes on pulmonary care. Are there any last questions about genetics anybody would like to ask? If not, you have a few minutes for a quick bio break. Get some dessert. There's delicious flan out there. Coffee or tea for anybody that likes. And I think at 1:15 is scheduled a session on pulmonary care.